Next question. Compare the rates of cell division occurring in your neurons and your hair follicle. Compare the rates of division occurring in your neurons and in your hair follicle. So it's very clear here for us to make this comparison. First, we are aware that actually the nerve cells enter or seem to stay permanently in the G0 stage, though some data suggests otherwise, which is still in question. And the reason as to why is because these cells rarely get worn out. Then we have these hairs which are growing continuously. The reason being that you can see that even after a shave, you won't stay long before the hairs will appear again. So you say neurons appear to stay permanently in the G0 phase. Neurons appear to stay permanently in G0 stage. So if they stay in G0 stage, it means they will not divide because they don't really wear out. Because they don't really wear out. So they are not wearing out, no need of replacing them. Then hair follicles divide more frequently. Hair follicles divide more frequently. More frequently. Otherwise, one would become bald. Otherwise, one would become one would become bald. Bald, by bald we mean he will not grow hair. But this also may depend in many other factors apart from actually division of cells. We know that some chemicals or some other factors may hinder growth. They may be pollutants and such stuff, or even age can reduce the rate of growth of hair. Neurons, actually, unless they are damaged, which is rare, they will not easily grow again. And because of this, many a times dangerous consequences are there if your neurons are actually affected. Next one is question 13. What is the relationship between a cell surface area and its volume. What is the relationship between a cell's surface area and its volume? So we know that actually an increase in a cell size increases also the surface area. So an increase in size, as a cell increases in size, its volume increases faster. So the volume actually increases faster than its surface area. So as a cell increases in size, its volume increases faster. Its volume increases faster than its surface area. Its surface area. Therefore, the further increase in size, therefore, a further increase in size a further increase in size could result in a surface area too small, could result in a surface area too small. Too small for the adequate exchange of materials. For the adequate exchange of materials. So materials would be difficult to be exchanged, and we know that actually for materials to be transported in adequate amount, there is a need for a surface area. Also, not just only the amount, with even the adequate speed to keep the inside of the cell functioning, because the cell needs these materials for its survival. In case it's not getting these materials, the cell might die. Next question, question 14. You know that a chromosome is a very long, continuous strand of DNA. How do proteins help condense chromosomes? So actually what happens here is that the DNA wraps around histones, proteins. DNA wraps around histone proteins. Similar to beads on a string. similar to beads on a string. Parts of the histones interact with each other also. Part of the histones interact with each other. Interact with each other, further compacting the DNA. Further compacting the DNA. So there are two things happening here. Histone to DNA wrapping and histone to histone interaction. So this is how actually the proteins help in, in continuously condensing the chromosomes. So question number 15. Describe what happens in each main phase of mitosis, that is prophase, metaphase, anaphase, and telophase. So you're supposed to describe what happens in each. So we will try to do that in a precise way. So first we will start with prophase. What happens during prophase? So the chromatin condenses into chromosomes. Chromatin condense into chromosomes. Chromosomes are actually more tightly coiled. Nuclear envelope breaks down. Nuclear envelope breaks down. Nuclear envelope breaks down. Nucleolus disappear. Nucleolus disappear. So you will not see these guys here. And centromeres and centrioles begin to migrate to opposite poles. Centromeres and centrosomes, it should be, not centromeres, centrosomes. Centrosomes and centrioles begin to migrate to opposite side. To migrate to opposite sides of the cell or pole. Side of the cell. Spindle fibers form from centrioles. Spindle fibers form from centrioles and radiate towards the center of the cell. That is as far as prophase is concerned. These are in brief, this is what happens during prophase. Next one, we go to metaphase. Metaphase. So during metaphase, spindle fibers attach to the structures on the centromere of the chromosomes. So spindle fibers attach to each chromosome. Attach to each chromosome. And align the chromosomes along the cell equator. And align the chromosomes along the cell's equator. Along the cell's equator, or the center of the cell. Next is anaphase. So let's see what happens during anaphase. So during anaphase, sister chromatids separate from each other. Sister chromatids separate from each other. The spindle fibers begin to shorten, which pulls the sister chromatids away from each other and towards the opposite pole. The spindle fibers begin to shorten. The spindle fibers begin to shorten, pulling the chromosomes to the poles towards opposite side of the cell. So this is what happens during anaphase. The next one, or the last, is telophase. So what happens during telophase? Telophase. A complete set of identical chromosomes is positioned at each pole of the cell. A complete So we'll stop here, then we'll come and complete this question in the next video.